Hello champs, welcome to my YouTube channel. Hope so you all are doing well. So today we are going to learn a topic that is transcription in just 10 minutes. So let's start our video. Transcription. What is transcription? Making DNA to RNA, or we can say transferring of information from DNA to RNA, is nothing but your transcription. How it happens, where it happens, and why it happens, let's see ahead. So transcription. Your transcription mainly takes place in the nucleus of the cell. So this is your nucleus. So in the nucleus of the cell, transcription takes place. Why? Because in your nucleus, DNA is present. And this DNA is having all the genetic information, right? And this genetic information must come out, or we can say to pass it on, the DNA makes an RNA and it transfers its information to the RNA. And talking more specifically, mRNA. And this process of transferring the information from DNA to mRNA is known as transcription. Okay, so as we are going to make DNA to RNA, the base pairs are also different. So talking about the base pair, our DNA has adenine, guanine, thymine, and cytosine, whereas your RNA has uracil instead of thymine. Let's talk about little structure of DNA before going to transcription. So this is our DNA. In DNA, we have, this is your 3' end, and this is your 5' end. This is your 5' end, and this is your 3' end. Okay, as your strands are running opposite, like one strand is 3 to 5 and the other is 5 to 3. This nature, because of this nature, we call as DNA is our anti-parallel in nature. And this one are your base pairs. This one are your base pairs: adenine, guanine, thymine, and cytosine. And talking more specifically about transcription, in transcription we use only single strand, only one strand of DNA. And this one strand of DNA which is getting used for transcription, this strand is known as template strand. Okay, template DNA, or we can say template strand. Okay, by seeing this template DNA or template strand, the mechanism is going to start. So for this mechanism, we have our hero over here, that is RNA polymerase. This RNA polymerase will study, or we can say analyze, our template strand, and according to it, it is going to add the nucleotides. Which nucleotide? Basically the nucleotides of RNA, as we are making DNA to RNA. Fine. For the transcription, we have 3 major stages or 3 major steps: initiation, elongation, and termination. So let's see all the steps in detail. Before going to each step, let me draw a structure. So here are main 2 regions. This region is known as promoter, and this region is known as terminator, and this is known as structural gene. This entire region with promoter, terminator, and structural gene is known as transcription unit. It is known as transcription unit. And the region which is getting we can say where the transcription is going on, that region is known as, we can say it is getting transcribed. That region is getting transcribed. Okay, and where the promoter is present, we call that region as upstream region, and the terminator region as downstream region. Okay, we have upstream and downstream. To talk more specifically, this was our 3' end and 5' end. This was our 5' and 3'. So this promoter is present at 5' end. Okay, let's elaborate it more now. So this was your promoter region, and this was your terminator region. Now comes the hero of our what we can say transcription. So here your RNA polymerase get binded. This is your RNA polymerase. it will get binded to promoter. Okay, it is a key player, we can say, of our transcription, who is going to make the RNA molecule by adding the nucleotides of RNA. Now this RNA polymerase will study, we can say, the template strand. So the strand that is a 3 prime to 5 prime, this is the 5 prime to 3 prime. The 3 prime to 5 prime here is your template strand. Now your RNA polymerase will go ahead and it is going to add the nucleotide. For example, if we have A, T, G, C, T, G, U, C. Okay. This A, for A we have base pair as U, for T we have A, for G we have C, and for C we have G, for T we have A, for G we have C. Okay, we have T over here, A over here, C, G, T. For T we have A, and for C we have G. So this is your promoter. This was your terminator. So what happens now? The RNA polymerase enzyme gets binded with the promoter over here, and it is going to run ahead, and by running ahead it is going to add the nucleotides. Okay, and this nucleotides are going to make your RNA. Let's see ahead. For example, these are your base pairs: adenine, thymine, guanine, cytosine, adenine, thymine, adenine and thymine, and these are their complementary base pairs. When this was your promoter, terminator, when your RNA polymerase is going ahead, it is adding the RNA base pairs. For A we have U, for T we have A, for G we have C, for C we have G, for A we have U, and for T we have A. If we look out here carefully, we can see that this ACG are similar as that of this strand. Only if we leave this U, rest of the things are same, right? That is why we call this strand as coding strand, because whatever your RNA is formed, it is similar to the coding strand and not that of the template strand. This template strand is also called as antisense strand, and this coding strand, it is also called as sense strand. Okay. So as this RNA polymerase is going ahead, it is adding the complementary base pairs of the RNA nucleotides, and here in this region your mRNA gets formed. And when this RNA polymerase reaches to the terminator, your transcription gets stopped. Transcription stops and your mRNA then gets released. So this was about transcription. Let's talk about it in just short again. We were having RNA polymerase as our key hero. Okay, what it is going to do? It is going to add base pairs according to RNA. Like RNA base pairs it is going to add. Whom he is going to read? He is going to read template strand. Where is your template strand? 3 to 5 prime end. Okay, so we were having one promoter region, one terminator region. Your RNA polymerase is going to bind to the promoter at 3 prime end, and now it is going to add the nucleotides. This nucleotide will be as per the RNA base pairing, and once this RNA polymerase reaches your terminator, transcription will get stopped, and this nucleotides will be similar to that of the template strand. It will be similar to that of the coding strand. This is your template strand, this is your coding strand. Okay, so this was all about your transcription. If you found the video is beneficial or has got you some of the knowledge, like, share the video, and please subscribe my channel. So that was for today. Thank you so much.